Hi, I'm Marvin Natowicz of the Cleveland Clinic, and I'm here today with Professor Stephen Scherer of the University of Toronto, and we're talking about genetic aspects of autism spectrum disorder, and we're continuing our earlier discussion. Because so many of the people who are being listening to us are practicing clinicians, can you distill for all of us what you believe are the key points from genetic perspectives that persons in clinical practice today, circa 2018, need to be aware of in terms of their practices?
So we did a survey about 5 years ago asking the families who are enrolled in our research, and there's over 3,000 of them across Canada, why they enrolled in our genetics research. And these were the three answers they gave us. The first was they wanted to know what the cause of autism was in their family member. They wanted to know, second, they wanted to know what they could do based on that information, if there was sufficient information to do anything. And the third was they wanted to help. They wanted to help other families. So those are the three driving questions that we form the answers to around to deliver the data back to families, to try to make sense of it. Okay, so I think when doctors suspect there's a diagnosis of autism, they should have a genetic test done. And the availability of a genetic test is going to depend on who the payer is, where you're at. As I said earlier, the chromosome test is the standard of care test now.
The chromosome microarray.
Chromosome microarray test. Some hospitals like ours still do the karyotyping too, because the healthcare system pays for that. But they do the karyotype, they do the chromosome microarray. It's going to move into exome and eventually whole genome. In the United States, it's mainly chromosome microarray now. But I think be prepared that it'll be exome very soon. There's a group of us getting together to make recommendations around this and probably later this year. So there's a lot of issues. So I think we try to answer questions and give the families more information. Okay, so what should you do to start? I think it depends on the scenario. If it's a male, if it's a female, if they are on a wait list, if there's a family history. We've written a series of reviews that have guidelines that essentially a decision-making tree based on those individuals. But I think you'd like to have the best test, the most genome-wide test available to you. So I would start with the chromosome microarray test. And that's also important because if something is found, that's what we have the most data on other families, of how they can actually use the data, what it means, what's the likelihood of recurrence, for example, what are the genes involved, that kind of thing. So I think that's the starting point, okay? And then wherever possible, try to get the families into research studies. There's a lot of ongoing research studies in North America where being in the studies and finding some of these genetic changes helps link them to clinical trials, for example, or to new information that they can enroll in dietary trials, for example, or changes perhaps in the medication that they're using for their seizures. All kinds of different changes in their lifestyle based on the genetic changes that they carry. And I think, certainly as researchers in the field, this is what we're trying to develop, is what the best care pathway is based on the genetic findings. But my recommendation is get the most comprehensive genetic test done as early as possible. Just to come back, in our province at any given time, there's actually in the city of Toronto, there's 500 children on a wait list to get a formal diagnosis from a developmental pediatrician of autism. So there's suspected diagnosis by a healthcare worker or a teacher. And that can take anywhere from a few months to a few years. As I said earlier now, the genetic tests we could prioritize. Like for probably 20% of the families, we would run the whole genome sequence in the best case scenario, or even the chromosome microarray test, and be able to prioritize a significant proportion, anywhere from 5 to 20% of these families, that they should be moved up the wait list. And then in addition, you should be looking at these high-risk individuals. So I think genetics will become more and more critical as we study more individuals, but we're seeing already that this is an important step in the workup. Genetics is never going to replace the behavioral diagnosis. But we've seen over and over and over again that actually can impact the clinical diagnosis and impact the families.
Of the examples that you've given so far, Professor Scherer, none of them, to the best of my knowledge, that are clearly associated with an autistic phenotype, whether it's a chromosomal copy number variant or a complete addition of a chromosome, as in, say, Down syndrome with chromosome 21, or in some of the single gene Mendelian conditions like tuberous sclerosis. And none of them do you see 100% penetrance of an autistic phenotype, even within the kindred, where one of the members of the kindred has the syndrome and is affected. Another person who has the syndrome may not have autism, but may have other manifestations of that syndromic condition. What are the current thoughts about why there is incomplete penetrance of a particular gene or genomic region that unquestionably is associated with autism?
Right. So just to come back to the twin studies, even if you have the exact same genome, and we can find a mutation in one of these high-impact genes in a very small proportion, maybe 10%, that's not sufficient to lead to ASD. So we know there's other factors involved. The genome encodes roughly 30,000 genes. About 25,000 of those encode proteins, and there's about 5,000 well-characterized genes that are called non-coding RNAs that are involved in a function in the cell. They just don't encode proteins. There's probably a lot more, but the ones we know about give us a total of about 30,000 or so. 75% of those genetic units, or we'll call them genes, are expressed in the brain. And the vast majority of those are specific to the brain. So the organ that's most influenced by genetics is the human brain, which is not surprising. The pancreas, just out of interest, is the one that has the least amount of genes expressed. So 75% of the genes are expressed in the brain. There are lots of targets for brain developmental conditions. Okay, and the brain is evolved to be a very fine-tuned, high-functioning, the most sophisticated, high-functioning, entity, unit, organ of anything that exists. So any aberrations can throw a wrench in the system. But at the same time, there can be other changes that can actually rescue the system. And of course, evolution is selected for these types of rescues. Many of these genes I've talked about come in gene families. So the SHANK3 gene, there's a SHANK2 and a SHANK1. The Norexin gene I touched on, there's at least 3 genes, and in all cases there's different, what we call isoforms of these genes. They're just different transcripts that overlap, or segments of the gene that overlap, that generate subtly different proteins. So they can rescue mutations that might exist in certain individuals. So this is what we call modifying. There can be a modification, a positive modification, that you're less likely to become autistic, or it's not a good way to phrase it, but it's a modifier gene. It can modify the outcome. And when we test, we're really only looking at one. We get the whole genome, but then we look at one gene at a time. So I think there's lots of ways to modify the outcome, and that's the actually really exciting thing here. If we can study those individuals that have these severe mutations that are doing really well, we can find out why they're doing really well. In a lot of the cases, it's a sex issue. It's the carrier mutation is actually female. So we published back in 2011 a very interesting family that had 4 generations, and they had a deletion of the SHANK1 gene on chromosome 19. This is an autosomal gene, and it was passed through 4 generations and only expressed as an ASD phenotype in the male carriers. And for some reason, the female carriers were protected. Their biology protects them. And this is not surprising, because we see a higher frequency in autism in males versus females, typically 4 to 1. So many of these genetic changes that we are studying are actually inherited. In fact, the vast majority of them are inherited. But for some specific regions of the genome, the mutations are always sporadic. Okay, so they're de novo in nature in that individual. So for example, at the very end of chromosome 3, chromosome 3q29, there's a genetic change that we see probably in 0.1% of autism or so. It's always de novo in nature. There's also another one on chromosome 2q31, which is always essential. I think every time we've seen it, it's a new genetic change in that individual, whereas most of the other ones can be inherited. And the reason that I forgot to say, is the clinical presentation of these individuals is more severe. Okay, so they tend not to be put in a situation where they pass on their genes. They have their own children. So of course their mutations aren't passed on. And then even to make it even more interesting, it turns out the genome carries these stretches of repetitive DNA that encompass about 5% of the DNA in the genome. And if they're spaced close enough together, they provide substrate for illegitimate recombination during meiosis and mitosis. So it's like when you're doing a zipper up and you get the zipper misaligned, and what happens is the DNA zipper misaligns and it deletes out these regions more often than not. So we see the same deletion event coming up over and over and over again. These are changes, and often they have a significant impact, and they're not passed on because the people don't pass on their genes. So what we're seeing again is autism is highly heterogeneous. You need to kind of be an expert on each specific genetic change you find. If it's a copy number variant on chromosome 16 or 22 or 3q or whatever it is, you almost have to be an expert on that region of the genome to make full sense of it. This is, I think, one of the challenges in the future for physicians is how to develop the best management or care pathway based on the genetic changes identified. And we've written a lot of reviews on this. In fact, this is an area that I find most interesting because we can now do the whole genome sequence from blood to sequence in less than a week. Find information from at least 20% of families that we're communicating back, and then it takes months to get the data back to the families because we have to develop the pathway to make sense of it and then deliver it back to the families through physicians. So we can talk more about that, but it's the future. This is what is called precision medicine or genomic medicine.
Now, in the lists of genes that are clearly associated with autism that are out there now, when one looks at those lists, there are a number of genes that encode structural proteins, like proteins of the synapse or various other membrane proteins. What one doesn't see a lot of in those lists are proteins involved in classic metabolic pathways. Do you have thoughts on that?
Right. So the approach we've taken is to scan the genome in a hypothesis-free manner, look for what are defined as high-impact mutations. So ones that we know either delete a copy of a gene or knock it out so that the protein's not made for that gene. So you may only have 50% of a particular protein versus 100%. In some cases, it goes up to an increase in the protein level through a copy number variant gain. But these are mutations typically that are kind of thought to be high impact in nature, and that it changes the copy number of the protein or the gene. Okay, then if you use database comparisons to group these into what we call gene networks or protein networks, and based on their function within the cell, you can start to see groups of proteins coming out. And I talked about the proteins that are working in neuronal communication and development of the dendritic spines, for example, how the neurons work. But we're only looking at really as a subset of those mutations. If you expand now into missense changes, so the just spelling mistakes in the genetic dictionary, if you will, you can look beyond that to see that there's other groups of proteins coming in. There are some known metabolic disorders that have autism associated with it. There's some mitochondrial mutations that are associated with autism. But generally, the groupings are falling into this synaptic protein. But I think we're looking under the lamppost. There's no question. We keep comparing to the same gene list. Okay, so we're now at a point where we have, as a community, the whole genome sequence from roughly 20,000 individuals. We'd like to get way above 100,000, maybe even 1 million or so. But with these tens of thousands of whole genomes, we have all the genetic variation. We can look at the protein coding genes and the non-coding genes. And in most cases, we've got the families too, so we can look at inherited mutations too and see if we can subcategorize the types of proteins that are involved. Right now, that data doesn't exist. We're going to find other protein networks involved. There's no question about it. And I think when we start to think about guiding the questions of how the environment may impact these proteins, I think that's the million-dollar question. Okay? Is it going to be that an individual that has no mutation in any of the genes that we've identified so far, if we look at their neuronal development, and we can do this now using induced pluripotent stem cells, do they have the same synaptic structure that we see in an individual that carries a SHANK3 mutation or an orexin-1 mutation? Is it really the neuron? We don't really know. We don't know what's going on for upwards of 75% of the families. But what we do know, as I said earlier, is, and we did know this a decade ago, is that the synapse is critical. Development certainly in some of these individuals.
So the reason that I asked the question about the listing of metabolic genes is many of the viewers of today's session are practicing clinicians, and they're dearly interested in treating their patients who have autism, who have significant issues that require treatment. And the means of treatment often are metabolic. They may be changing a diet, giving supplements, providing medications, but augmenting one or more metabolic pathways in order to influence the cellular and tissue phenotype. And so do you have hope that with time, through mining of the data, through such techniques as whole genome sequencing, there will be opportunity to have impact on metabolic pathways that will be amenable to types of manipulations that clinicians do to help patients?
I think it's going to be possible. So Art Baudet from Houston has done a nice study where he looked at the entire genome, but he focused on the X chromosome and metabolic genes, and he's shown in a very, very small proportion of individuals, males that have ASD, a metabolic defect. And I think there's other clues. We've looked specifically at metabolic genes by both direct sequencing and the whole genome. We don't see a major factor, but that doesn't mean that existing metabolic treatments are not going to work. I think we have to take these protein networks one step further, okay, and then link the whole genome sequencing, the barcode of their genetic information, to clinical trials. If there's a good hypothesis based on the clinical presentation or rescue of it in, say, a small group of individuals, it may have a broader impact. This is the next area of development where you take the genome sequence and then look at how the gene products, the transcripts, the RNA at the RNA level, for example, are regulated. Okay, you might get some common patterns there that link with metabolic disturbances, for example, and can be treated. But I think right now, it's very, very rare. There's no single gene that's been identified, or CNV that accounts for more than 1% of autism. And then if you tie together the genes that are involved in the synaptic function, for example, you get you can account for upwards of maybe 5% of the family. So there's still a gap to be found. And I think it's very, very early days. It may very well be that metabolics is the best way to go. We just don't know yet.
Might it be possible that even though on the current listing of genes clearly associated with autism, while there isn't strong representation of any metabolic associated genes, might it be possible that some of the known genomic lesions can secondarily impact one or more metabolic functions, say, for example, mitochondrial bioenergetics, and that might be a common theme and also potentially amenable to pharmacologic or nutritional manipulation and treatment.
Yeah, that is certainly possible. I think as what you were asking earlier, is you need as much deep phenotype data as possible to drive some of the hypotheses in the genomic investigation. The beauty of the whole genome sequencing is, as I said earlier, it's really good data. It's essentially static. I don't think we're going to be doing this again for a long time. We've only been really doing association studies looking at these high impact mutations, but if you can go and look at missense changes and other types of genetic changes, there may be a correlation there to be found. Okay, but you need to have the deep phenotype data. In this case, it's metabolics. I think moving the testing into the clinic, the copy number testing, the chromosome testing that's done now, is not going to find these types of changes that I'm talking about. So we need to evolve it into the exome and the whole genome sequencing. And I think you'll see through numbers some of these associations start to fall out. But let's wait and see.
Several times you've mentioned what is already apparent from the data that we have in terms of the importance of mock-ups of synaptic biology in terms of autism pathobiology. Are you comfortable at this point endorsing any other biological themes in the pathobiology of autism as strongly or nearly as strongly as you've endorsed mock-ups in synapse biology?
I think that's our best hint and best clue right now. The data supports dysregulation of synapse biology as the primary route based on the data that exists. But again, it's very early days. I think everything's changing every time a new study comes out, but everything so far is mapping back to that converging theme. Where there's dysregulation in homeostasis of these proteins. And there was a very nice study done. So let's just backstep for a second. So, of course, the challenge of doing autism research is you don't have direct access to the brain cells, right, to the brain of the individual. So there's a technology called induced pluripotent stem cells that allows you to take a skin fibroblast or a blood cell or even urine now and establish through the induced pluripotent stem cell technology stem cells that can then be differentiated into any tissue in the body, including the brain. Okay, so we can develop neurons from that individual that carries a specific mutation. And in the culture dish, these can be grown. In some cases, you can even now turn them into what's called a brain organoid, to have a little miniature brain. And you can test them both based on histochemistry, electrophysiology, to see what they look like, how they're performing. And in those mutations that have been modeled, and there's been probably 50 different mutations published on now, including many from our group. We see, now these are genes like the neurexins in the shanks, that's why we're modeling them. You see changes in the density of the dendritic spines, for example, the length of dendrites, the number of neurons, that kind of thing, structural changes, as you said earlier. There was a really nice study published where they took iPS-derived neurons from individuals that didn't have any mutations and looked at them closely and found a lot of the same changes. Okay, they only looked at, I think it was 10 individuals, but suggesting that the same genetic pathways will be involved in those people who don't have these mutations. It's just scratching the surface. I'm sure there's going to be lots of variation, but I think that's one of the big questions in the field. Can you develop a technology as a proxy or a sensor for those non-genetic forms, or at least the forms we haven't yet found to be genetic, and see if they have the same impact on the brain? Now again, we're ascertaining, we're only looking at these neuronally derived culture lines in the tissue culture plate. There could be something else going on here we haven't done the association test for, like metabolics you said earlier. And we have lots of clues from these rare forms of ASD that metabolics and infections can be involved. But this is the best data so far, and I think it's data we didn't have 10 years ago. It allows us to start to develop models to see what else is going on. Mouse models, for example, where you can look at all of the organs in the animal.
And while none of us have a crystal ball, what does your crystal ball tell you in terms of things that clinicians need to be watchful for in the near horizon?
Well, so there's a lot happening now. Genetic testing is just going through the roof, and as you move towards whole genome testing, you get data on all 30,000 genes, right? So we're often seeing that a parent or even a child or even a developing embryo is tested using a whole genome test. And it may be for question X, but you find a genetic change in one of these autism risk genes I talked about. So how do you deal with that information? So that's coming. I bet you most of your viewers have had that experience already. Certainly I get an email every single week or almost every day now from around the world because we publish a lot of these papers is, what does this result mean? So I think it's very important to say there's no single gene identified or CNV identified, copy number variant identified, that is entirely predictive for an autism outcome. And we don't even know, even if we were able to predict, and there's a family history of autism, where on the spectrum that individual will land, right? Because again, in monozygotic twins, in about 15% of cases where there is concordance, one of the twins is severely autistic and the other is Asperger's syndrome, for example. So this is broad range of clinical presentation. But there is some data that can be had. So I think prepare for those complex questions. What we've done and what we're recommending, and I think it's the same in most places in the States, is to have a genetic counselor involved who's helping to interpret this data because it is so complex. I gave a few examples of of the chromosome 16p and 22q having different recommendations. There's hundreds of these genes, and it's going to be very hard to stay up on top of this unless you're the expert in the field. So you have to rely on genetic counselors. With these direct-to-consumer testing companies really, again, catching a lot of interest in North America, there's going to be more findings coming back that are going to hit autism genes. So you need to know what to do with the data. I think that will be the number one thing. The other exciting advance is that now that we've identified some of the gene targets, and it's only been in the last decade, there are new testing going on and development of new medicines, for example, to modulate these genetic pathways. So I think if you have found a family member who has autism and you can identify their mutation, you should look in research databases to see if there are any clinical trials that are ongoing on a research basis that these individuals may enroll in. NIH has got a list that you can go and look at on their website. And there's an interagency coordinating committee of NIH that is working to get all of this data together. So I think use web resources is really, really critical. The families are great at this. They're going to be bringing the data to you too. So I think kind of have a process in place of how to deal with the multiple types of information coming to you. We've now found, I think it's been interesting from a genetic perspective. Autism was a behavioral defined disorder. And I mentioned that the majority of all of the genes we found are involved in brain development, but probably half of them have another function too, okay, in some other multi-system function, so in heart development or in limb development. And this list continues as we know more and more about these genes. So I would say in about 50% of cases, when we're writing the clinical report on these genetic changes we're finding in 20% of the families, they're being referred to multiple clinics, not just going through clinical genetics. So I think be prepared for an onslaught of information. And the best way to interpret these results is using a multidisciplinary approach. It's not just going to be the case for autism. There's convergence in many different disorders. It's interesting, I think, to subdivide these different groups. That's the real emphasis for our whole genome sequencing project. But there's going to be a lot of information, so you need to get information savvy.
And you've been involved for a very long time now at the forefront of autism genetic research. In your mind, what do you regard as some of the most exciting findings in recent years that you've been witness to?
I would say everything's come in the last 10 years. That's really, really exciting. And that in itself is exciting. So the first is that a decade ago, we didn't have any genes for the so-called idiopathic or essential autism at the time. And now we have a list of 100 or so targets that we can start to think about, both use in a diagnostic setting to answer some of the family's questions that are brought to the clinic, and then also design experiments using these iPS-derived neurons, animal models of mutations, to test either existing drugs or develop new drugs to these known targets. I think this is absolutely mind-blowing. We've moved from understanding autism from a behavioral basis to having, in some cases, specific genes we can develop rational experiments around. That's changed everything. And in some cases, people have done this already. They've actually taken a human mutation, modeled it in a mouse, get a similar behavioral presentation in the mouse that looks like a mouse form of autism. The animals don't interact as much, or they're highly anxious, and there's different assays that have been developed to test this. And then based on the genetic pathway that's affected, you do a replacement of a particular protein or some type of a drug that modulates that protein, and you can rescue the mouse phenotype. So you can actually reverse the autism in there's been some studies in Shank3 mice, for example, and Neurexin mice. So the developmental process can be reprogrammed in mice, okay? So maybe that's going to be the case for human too. I think there's a lot of hope that exists now that we can actually, in an informed way, develop new medicines to actually modulate these chemical processes in the brain for autistic individuals and offer that as an answer to the families. We hear back from many of the families now that have been in our study for decades. We started this probably 30 years ago. Prior to when I started my laboratory, there were clinicians collecting these wonderful families across Canada and in the United States, and the siblings of these first index cases where we collected are now growing up and thinking about having their own families, and they want to know about genetics. So again, the fact that we can start to give them some answers is really, really exciting. And I think it's going to just snowball. Every single time we go back and look, I should have said this earlier, when we took about 1,000 genome sequences now from clinical genetics. Many were autism. It wasn't entirely an autism study. But we did the whole genome sequencing on roughly 1,000 families. And a year after the initial analysis, we analyzed it again and compared to all the new data that was coming from all different studies from around the world. And we increased the clinical yield by 10%. So every time we look at the data, we learn a lot more. So I personally feel that in the last decade, probably in the last few years or so, we've kind of turned the corner where we're starting to see a convergence, we're starting to get some answers, and we're just getting a lot smarter about the science we're doing. And I think it's going to be very similar to what we saw in the decade of cancer. We see treatments now coming for specific forms of cancers based on genetic findings. We're probably about a decade behind. I think we'll see that into the next decade for autism.
One final question for you. What do you, having worked in the field so long and so hard and done so much, what do you feel are the most important still unanswered questions that need attention?
Yes, I think I'd like to do several experiments. The first is that I would like to just sequence every individual that has a neurodevelopmental diagnosis. I probably didn't emphasize it enough in my earlier comments that every single autism gene we've identified, again, as a community, is found in other neuropsychiatric and neurodevelopmental disorders. Okay? So ideally, we would remove the autism diagnosis and just sequence everyone first and see which individuals have specific mutations, and then go back and do deep phenotyping on them, and then try to reconstruct the definition of these behavioral neuropsychiatric neurodevelopmental conditions. I think for a subgrouping, you're going to see, and we already have data for this, you're going to see an entirely different grouping of genetic disorders. Very interestingly, the Simons Foundation funded this great study where they looked at families that had one affected individual. And in the early days, they found these chromosome 16 changes, which now are identified using chromosome microanalysis, and they're identified as chromosome 16 forms of autism. And they don't go into research studies anymore. So you don't find those anymore if you're to do a research study. Okay? So you have to group based on genetics and phenotypes. I think that's starting to happen. It just needs to be coordinated in a way. And then also, probably most importantly, is to bring new people into the field who have completely new ideas and new entry points. And from my perspective, I think the biggest set of answers are going to come using what are called machine learning, artificial intelligence-based programming, looking for patterns in both phenotypes and in the genotypes. I work in the genotypes. We're already using these. We're finding specific mutations now using what's called deep learning approach that we couldn't identify using traditional computer algorithms that we've developed using other approaches. So, but we need to have high quality, large data sets to do this. And we published a paper a few years back already now, found specific mutations in how the RNA splices only using artificial intelligence approaches, and you can't find it using other approaches. So when we get all this data together and we use AI approaches, I think we're going to find some completely new observations that are not looking under the lamppost, these kind of outliers that start to tie things together. That's what I'm really excited by, to get our numbers up and to use AI and to see what else we can find to find answers for the families.
Well, thank you so much for sharing your insights with us today.